In today's lecture, let us look at the classification of proteins, one of the most important biopolymers. Proteins may be classified according to various criteria such as chemical composition, shape, functions, solubility, structure, and nutritional value. Let us now look at the classification based on chemical composition. Depending upon their chemical compositions, proteins may be categorized into three types. Simple proteins. These proteins are also called homoproteins. They consist only of amino acids. For example, albumin, collagen, keratin, globulin, etc. Based on their differential solubilities in water, organic solvents, and salt solutions, simple proteins are classified into several types such as albumins, globulins, prolamins, glutealins, protamins, histones, and albuminoids, etc., which we shall see in detail later in this presentation. The second type of proteins based on chemical composition are conjugated proteins. They are also known as heteroproteins. Upon hydrolysis, they yield amino acids as well as non-protein components called prosthetic groups. These prosthetic groups could be metal ions, phosphate groups, or they could be pigments, lipids, nucleic acids, and flavins. They may be subclassified into mucoproteins and glycoproteins, chromoproteins, phosphoproteins, nucleoproteins, lipoproteins, flavoproteins, and metalloproteins. Now, let us discuss the category called mucoproteins and glycoproteins. Mucoproteins and glycoproteins have one or more carbohydrate units attached to the polypeptide backbone. Typically, the branches consist of 15 to 20 carbohydrate units, which could most frequently be arabinose, glucose, galactose, fructose, mannose, N-acetyl glucosamine, and N-acetyl neuraminic acid, NANA. Glycoproteins contain smaller amounts of carbohydrates, say about less than 4%, whereas mucoproteins contain comparatively higher amounts, more than 4% of carbohydrates. Examples of glycoproteins include glycophorin, fibronectin, and all plasma proteins except albumin, immunoglobulins, and antibodies. Some well-known mucoproteins are ovomucoid, present in egg white, mucin present in saliva, osteomucoid present in bone, and tendomucoid present in tendons. The next group is chromoproteins. Proteins with colored prosthetic groups are known as chromoproteins. Examples of such proteins include hemoglobin, retinal, myoglobin, hemocyanin, hemoerythrin, cytochromes, flavoproteins, and catalase, etc. Chlorophyll is present in leaf cells in the form of a protein called chloroplastin. The next group is phosphoproteins. These are proteins with phosphate groups attached to serine and threonine residues. Examples of such proteins are caseins present in milk, ovovitalin present in egg white, and phosphovitin. Nucleoproteins form the next group. These are basic proteins, for example, histones and protamins complex with nucleic acids. They are present in the nucleus as well as cytoplasm. These may be considered as the sites for the synthesis of proteins and enzymes. Examples include nucleoproteins in yeast and thymus, and also viruses, which may be regarded as large molecules of nucleoproteins, and the nucleohistones from nuclear-rich materials like glandular tissues, nuclein, etc. Lipoproteins form the next group. These are proteins forming complexes with lipids such as cephalin, lecithin, and cholesterol. They are soluble in water but insoluble in organic solvents. Examples include lipovitalin and lipovitalenin in egg yolk and lipoproteins present in blood. Lipoproteins are actually temporary intermediates in the process of transfer of lipids from the site of absorption to the site of utilization. The classification of lipoproteins is often based on the extent of migration of the lipoprotein fraction in a density gradient separation. Thus, the lipoproteins have been further classified into 4 categories: very high density lipoproteins, VHDLs. These have densities greater than 1.21. High density lipoproteins, HDLs, with density range of 1.063 to 1.21. Low density lipoproteins, LDLs, their densities range between 1.05 and 1.063. And the last category is very low density lipoproteins, VLDLs, with density ranging from 0.93 to 1.05. The next group consists of flavoproteins, which are proteins that contain a nucleic acid derivative of riboflavin, the flavin adenine dinucleotide FAD or flavin mononucleotide FMN. Examples include flavoproteins present in the electron transport chain in the mitochondria. The next group is metalloproteins. These are the proteins linked with various metals. These may be of stable nature or may be more or less labile. Based on their reactivity with metal ions, the metalloproteins may be classified into 3 groups. Metals strongly bound by proteins. The second group could be metals which are weakly bound by proteins. Calcium belongs to this group. The next group are those metals which do not couple with proteins. Sodium and potassium belong to this group. Derived proteins are proteins derived from simple or conjugated proteins by the action of heat, enzyme or chemical reagents. They could be of 2 types: primary derived proteins and secondary derived proteins. Primary derived proteins. These are derived proteins whose sizes do not differ substantially from the parent molecules. These primary derived proteins could again be of various subtypes, such as coagulated proteins, proteins, or they could be metaproteins. Now, let us look at secondary derived proteins. These are derived from the parent proteins through hydrolysis and are therefore smaller than the original proteins. They could again be of various subtypes, such as proteases. Proteases are soluble in water and are coagulable by heat. They are produced when hydrolysis proceeds beyond the level of metaproteins. Examples of proteases include albumose, derived from albumin, and globulose, derived from globulin. The next category is peptones, which are soluble in water and non-coagulable by heat. Peptones are produced by the action of dilute acids or enzymes on proteins when hydrolysis proceeds beyond proteases. They are neither salted out by ammonium sulfate nor precipitated by nitric acid or picric acid. Polypeptides form the next group of derived proteins. These are chains of 2 or more amino acid units. In fact, the proteins are essentially long chain polypeptides. The next category is peptides. Peptides are short polypeptides, but the distinction between polypeptides and peptides is rather arbitrary, and there is no clear-cut demarcation between the lengths of peptides and polypeptides. Now, let us look at classification of proteins based on shapes. Based on their shapes, proteins may be classified into 3 groups: fibrous proteins, globular proteins, and intermediate proteins. Fibrous proteins. These have axial ratios greater than 10 and henceforth resemble long ribbons or fibers in shape. These are mainly of animal origin and are insoluble in all common solvents such as water, dilute acids, alkalis, and salts, and also in organic solvents. Most fibrous proteins serve structural or protective roles. Now, let us look at the next category, that is globular proteins. These proteins have an axial ratio, that is the ratio between the length and the width, of less than 10, usually not over 3 or 4, and henceforth these proteins possess a relatively spherical or ovoid shape. These are usually soluble in water or in aqueous media containing acids, bases, salts or alcohol and diffuse readily. Intermediate proteins. Intermediate proteins have axial ratios lesser than that of fibrous but greater than based on their functions. Proteins may be classified into various groups such as catalytic proteins, which are mostly enzymes. The most varied and highly specialized proteins are the enzymes. Almost all the chemical reactions of the body are catalyzed by these catalytic proteins. Urease, amylase, catalase, cytochrome C, alcohol dehydrogenase are some of the examples of enzyme proteins. The next category are transport or carrier proteins. Certain proteins, especially in the animals, are involved in the transport of many essential nutrients or gases to various parts of the body. For example, hemoglobin binds oxygen as the blood passes through the lungs and carries it to the peripheral tissues and releases it there to participate in the oxidation of nutrients. Lipoproteins present in the blood carry lipids from the liver to other organs. Other kinds of transport proteins are present in the plasma membrane and intracellular membranes of all organisms. Ceruloplasmin transports copper in blood. Storage proteins form the next class. These proteins store important nutrients or essential elements, for example, phosphorus, calcium, and iron. Ovalbumin is the major storage protein of egg white. The milk protein casein stores amino acids and phosphorus. The seeds of many plants store nutrient proteins required for the growth of the germinating seedlings. Ferritin found in some bacteria and in several plant and animal tissues stores iron. Structural proteins form the next category. Structural proteins usually do not undergo biochemical reactions. They maintain the native form and position of the body and organs. The next category is motor proteins. Some proteins endow cells and organisms with the ability to contract, to change shape, or to move about. Actin and myosin function in the contractile system of proteins form the next group. Some proteins mediate the regulation of cellular and physiological activities. This group includes many hormones such as insulin, which regulates sugar metabolism, and growth hormone, which is required for bone growth. Defense proteins form the next type. Many proteins defend the organism against invasion by other species or protect them from injury. The antibodies or immunoglobulins, the specialized proteins made by the lymphocytes of vertebrates, can precipitate or neutralize invading bacteria, viruses, or foreign proteins from another species. Fibrinogen and thrombin are blood clotting proteins that prevent the loss of blood when the vascular system is injured. The next group is receptor proteins. These proteins mediate signal transduction, acting as binding sites for ligands such as hormones. Examples are receptors for peptide and steroid hormones. The next category is pigment proteins. For example, rhodopsin proteins with cofactor called retinal that are involved in vision. Some proteins act as toxins, protecting the organisms which produce them. Examples include snake venom, bacterial toxins and toxic plant proteins such as heparin and ricin. Nucleoproteins form the next group. These proteins associate with nucleic acids and regulate their functions. For example, capsid proteins of viruses, histones in chromatin, and ribosomal proteins in ribosomes. The next category is special proteins. These are proteins with special functions, and we will not deal with them in detail here. Designer proteins are proteins specially designed. Simple proteins may be subclassified into various subtypes. That is, albumins. Albumins are readily soluble in water, dilute acids and alkalis but coagulated by heat. Globulins. Globulins are insoluble or sparingly soluble in water, but their solubility is greatly enhanced by addition of neutral salts such as sodium chloride. The next category is prolamins. Prolamins are insoluble in water but soluble in 70 to 80% aqueous alcohol. Upon hydrolysis, they yield high amounts of proline and amide nitrogen, hence the name prolamin. Glutalins form the next group. Glutalins are insoluble in water and absolute alcohol but soluble in dilute alkalis and acids. They are usually plant proteins, for example, glutenin present in wheat. The next category is histones. Histones are small and highly basic proteins. They contain high amounts of basic amino acids such as histidine, lysine, and arginine. Protamins form the next category. Protamins are simple proteins which are soluble in water and are not coagulated by heat. They are basic in nature due to presence of large amounts of arginine. Let us look at the next category, that is albuminoids, which are also known as scleroproteins. These proteins are characterized by great stability and insolubility in water and salt solutions. They are called albuminoids as they are essentially similar to albumin and globulins. Next, let us look at protein classification based on their structures. According to the structural classification of proteins, that is the SCOPE system, proteins may be structurally classified into various classes based on the types of structural folds they contain. Now look at classification based on nutritional value. Based on their nutritional value, proteins could be classified into three types, that is complete proteins, incomplete proteins, and complementary proteins. Complete proteins. A complete protein is one that provides all of the essential amino acids. You may also find these sources called as high quality proteins. Animal-based foods, for example, meat, poultry, fish, milk, eggs, and cheese are considered complete protein sources. Incomplete proteins. An incomplete protein source is one that is low in one or more of the essential amino acids. For example, proteins derived from rice or beans. Next category is complementary proteins. Complementary proteins are two or more incomplete protein sources that together provide adequate amounts of all the essential amino acids. Today, we had a detailed look at the classification of proteins. We hope that you must have had a good understanding of how proteins are classified based